Welcome to the Explainer. Grab your lab coats because today we're stepping right into the role of plant doctors. Whether you've got a massive biochemistry exam coming up or you just really want to master the hidden, complex world of plant nutrition, you are in exactly the right place. We're going to decode exactly how plants take these invisible, inorganic elements straight from the earth and transform them into the life-sustaining biological structures that we literally rely on every single day.
Okay, let's dive into this. Here's our agenda for today. Section 1 covers the rules of essentiality, Section 2 is macro and micronutrients, and Section 3 is diagnosing deficiencies. Then Section 4 tackles uptake mechanisms. Section 5 breaks down the nitrogen cycle, and we'll wrap it all up with Section 6, the exam cheat sheet.
Section 1. The rules of essentiality.
So to really get this system, we first need to lay down the absolute basics. Mineral nutrition is basically the process where plants absorb and utilize essential inorganic elements from the soil for their growth, development, and metabolism. I want you to picture plants as nature's ultimate biochemical factories. They don't just, eat dirt. They're highly specific. They pull inorganic ions straight out of the earth via their roots, absorbing nitrogen as nitrate or ammonium and extracting phosphorus and potassium in their purely ionic forms. They pull all of this up to fuel these incredibly complex organic assembly lines.
But hey, not just any random element gets VIP access to this factory. Back in 1939, two scientists, Arnon and Stout, established the ultimate biological ground rules. So, the crucial point is, an element is only truly essential if it meets 4 very strict criteria. First off, the plant literally cannot complete its life cycle without it. Second, if it's missing, you are guaranteed to see abnormal growth. Third, it is completely irreplaceable, meaning you can't just swap it out for a similar element and hope for the best. And finally, it absolutely must be directly involved in the plant's metabolism. If an element doesn't tick every single one of those boxes, it's just not essential.
Section 2. Macro and micronutrients.
All right, if you want to effortlessly memorize these elements for your exam, consider these mnemonics your absolute cheat codes. For the macronutrients, which are the ones the plant needs in large amounts, just remember CHON plus NPK plus CMGS. That gives you carbon, hydrogen, oxygen, nitrogen, phosphorus, potassium, calcium, magnesium, and sulfur. For your micronutrients, the ones needed only in tiny trace amounts, just memorize this exact rhythm. FeZn-Mn-Cub-MoCl-Ni. That covers iron, zinc, manganese, copper, boron, molybdenum, chlorine, and nickel. Seriously, jot these down right now. They're going to save you on test day.
To understand why we even split them up this way, you just have to look at their jobs. Think of your macronutrients as the heavy lifters. Carbon and nitrogen are literally building the physical structures and the DNA. Phosphorus is handling all the energy transfer, like ATP, and magnesium, it sits right at the very heart of the chlorophyll molecule to make photosynthesis happen. Contrast that with your micronutrients. These guys are the precision tools. Zinc, for example, is vital for synthesizing plant hormones like auxin. Copper is a major player in electron transport. They're only needed in trace amounts, sure, but without these microscopic enzyme activators, the entire heavy-lifting machinery just completely grinds to a halt.
Section 3. Diagnosing plant deficiencies.
Have you ever looked at a struggling houseplant in your living room and noticed something kinda odd? Usually, the oldest leaves down near the bottom turn yellow and die off first, while the brand new baby leaves at the very top look perfectly healthy and green. How does that even make sense? Why wouldn't the whole plant just suffer equally?
Well, the answer lies in nutrient mobility, and this brilliantly illustrates the ruthless survival mechanics of the plant world. Certain nutrients, like nitrogen, phosphorus, potassium, and magnesium, are what we call mobile. When a plant senses a shortage in the soil, it literally cannibalizes its older leaves. It packs up those mobile nutrients and ships them upward to prioritize the survival of its new growth. That is exactly why older leaves show symptoms first. On the flip side, you have immobile nutrients like calcium and iron. Once these get built into plant tissue, they are locked in place. They cannot move. So if the soil runs out of calcium, the older leaves stay nice and green, but the new growth comes out all yellow and distorted.
Okay, so when you're diagnosing these deficiencies on a test, or even just out in a garden, you need to recognize these exact symptoms. First is chlorosis. This is that classic yellowing of the leaves caused by a loss of chlorophyll, and it usually points to a lack of nitrogen, magnesium, iron, or sulfur. Next is necrosis. This is the actual death and browning of plant tissue. Think crispy brown leaf edges, which signals a severe lack of potassium, calcium, or magnesium.
Now, if you see deep, dark purple leaves, you are almost certainly looking at a phosphorus deficiency. And finally, if the bottom of a fruit is just rotting away, a condition we call blossom end rot, you instantly know that plant is starving for calcium.
Section 4. Nutrient uptake mechanisms.
Under the soil, everything, and I mean everything, starts with root hairs. These are the microscopic heroes of the whole biosystem. By projecting outward into the surrounding soil, these tiny root hairs dramatically increase the overall surface area of the root system. And that massive surface area has one primary job: absorbing water and soaking up all those vital mineral ions we've been talking about. They just totally maximize the plant's physical contact with its environment.
But how do those ions actually get inside the root once they're found? Well, it happens in two main ways. First is passive transport. Think of this as an effortless glide. It requires zero ATP energy from the plant because ions are simply moving along the concentration gradient, basically just diffusing from areas of high concentration out in the soil to lower concentration inside the root. But a lot of the time, the plant really needs nutrients that are super scarce in the soil. That is where active transport comes in. This is an energy-hungry uphill battle. The plant actually has to burn ATP to power specialized carrier proteins. These proteins literally force ions against the concentration gradient, ripping them from the soil and pulling them in, even when the soil is holding onto them tightly.
Section 5. The complex nitrogen cycle.
Let's move to and see how this builds step by step, because nitrogen is a fascinating puzzle. Nitrogen is literally everywhere. It makes up around 78% of the air we're breathing right now, and yet plants can't use a single drop of it in its gas form. So step 1 is that atmospheric N2 gas. Step 2 is nitrogen fixation, where that useless gas is pulled out of thin air and converted into ammonia. Step 3 is nitrification. This is a crucial soil process that converts ammonia first into nitrite and then into nitrate, which is the form plants actually prefer to drink up. And finally, step 4 is plant uptake and assimilation, where those nitrates are absorbed by the roots and built right into the plant's own proteins and DNA.
But here's the catch. Plants absolutely do not do this alone. They rely entirely on a highly specialized invisible bacterial workforce. Biological fixation? That's driven by bacteria like Rhizobium and Azotobacter. Nitrification, that super crucial conversion step, is powered by Nitrosomonas and Nitrobacter. And completing the whole cycle by releasing nitrogen back into the air, a process known as denitrification, is handled by Pseudomonas bacteria down in anaerobic soils. Seriously, if you're taking a biochemistry test, you must know these bacterial names and the specific jobs they're doing.
Probably the most elegant example of this teamwork is symbiotic nitrogen fixation. This is famously seen between Rhizobium bacteria and the roots of legume plants, so think peas and beans. The plant actually goes out of its way to build specialized little root nodules just to house these bacteria. And if you cut one open, inside these nodules, you'll find a remarkable pink pigment called legumoglobin. Its job is completely fascinating. You see, the specific enzyme that fixes nitrogen, which is called nitrogenase, is totally destroyed by oxygen. So legumoglobin acts as this incredible oxygen shield. It binds up the free oxygen to maintain a perfectly low oxygen environment, which keeps the nitrogenase enzyme active and the nitrogen flowing. It's just a brilliant mutualistic relationship.
Section 6. The exam cheat sheet.
First up for the cheat sheet, you've got to be ready to define hydroponics. This is the technique of growing plants in a highly controlled nutrient solution, completely without soil. By using hydroponics, scientists can intentionally withhold one single specific nutrient to precisely study those exact deficiency symptoms we covered a few minutes ago. It's a highly testable vocabulary word, mostly because it perfectly leverages all of those rules of essentiality we talked about, but in a totally controlled soil-free environment.
Now, what's really interesting about this slide is that these enzymes basically represent guaranteed points on a biochemistry test. You really need to know these 4 players. Nitrogenase is that delicate, oxygen-hating enzyme driving the initial nitrogen fixation. Once the nitrate actually gets inside the plant, nitrate reductase steps up to convert it into nitrite. Then nitrate reductase converts that into ammonium, so the plant can start building amino acids. Finally, urease is absolutely essential for breaking down urea. And hey, remember our micronutrients from earlier? Urea specifically requires the micronutrient nickel to even function.
Which leaves us with a pretty profound realization to wrap up our explainer today. We've seen just how perfectly orchestrated this whole biological machinery really is, from mobile nutrients literally rushing upwards to save young leaves, to legumoglobin carefully shielding delicate enzymes from oxygen.
But just think about this for a second. If a single microscopic enzyme failure in the dirt, or even just the lack of one trace element like molybdenum or nickel, can completely halt a plant's ability to process nitrogen and build proteins, just how fragile is the global food chain we all rely on? The sheer brilliance of mineral nutrition is breathtaking, but its delicate balance is something every aspiring plant doctor has to respect. Thanks so much for joining me for this study session, and hey, good luck on your exams.